Tonight, as Peter mentioned, I'm going to kind of introduce you to the technology that we've developed and used over the last 30-some years for identifying triggers of inflammation in the form of foods and chemicals and food additives, herbs, and various environmental substances and so forth. You can imagine it's a broad subject because in the literature, which began quite a long time ago, adverse reactions to foods can impact any part of the body because the immune system permeates the entire body. And when the immune system gets activated through various mechanisms, it can, in the process of trying to restore balance or defeat a perceived threat, can cause bystander damage. But at the same time, after the damage is done, it's involved in restoration of homeostasis. So it's a very, very complex system. And we take a snapshot of it. We look primarily at the peripheral immune cells in the whole blood. And I'll get into some explanation of the technicalities of how we do that with the assistance of some graphics, which makes it easier. And as you see tonight, we want to talk about celiac versus non-celiac gluten sensitivity, because that's a big topic and there's quite a bit of confusion about it. Aside from gluten, there's also any food or other substance can be a trigger of innate immunity, and particularly we'll focus a bit more on neutrophil activation because it's the innate immune system, even though the specific immune system is represented in what we test and can respond. The way our assay is set up, it's primarily the innate immune system that we see becoming activated. And of course, leave time for questions. So basically, food intolerance became more talked about in the 1950s and 1960s after an allergist in Chicago named Theron Randolph discovered that a lot of vague symptoms that people presented with that didn't fit a discrete diagnostic category were actually triggered by environmental substances. And he discerned that by taking extremely careful notes during the history of a new patient, which might go on for about 3 hours. And one of the things he observed was that the symptoms were typically chronic and low grade, or medium grade. They tended not to be acute the way symptoms present with a classical type 1 allergy. And in addition, chronic exposure to the food or the chemical, perhaps in the workplace, would tend to mask the symptoms. So he found, for example, that if there was a person involved in the printing industry, and during the work week, he might be sort of okay. And then during the weekend, his body would start to ache and get headaches and couldn't wait to get back to work. He realized that what the exposure was doing was sort of masking the symptoms, and that when the exposure was lifted, there was a certain phase of demasking, which could be very uncomfortable. But what that also meant was that identifying the trigger was not so easy. So for example, in contrast to a classical allergy, which is also in the Gell-Coombs systems called a type 1 allergy, which we've known to be mediated by IgE, the symptom onset is clearly associable with the ingestion of the food or the exposure to the trigger. So if someone eats crustacean and their throat closes up within moments, it's clear. But if somebody has these chronic ongoing symptoms that are not so acute, they're maybe more vague, as I mentioned, they can impact any part of the body, identifying what causes that is more difficult. And so there was a number of attempts that were made to objectively try and identify these triggers. And in the 1930s, there were a pair of let me back up even, no, let's skip over that. In the 1930s, there was a pair of allergists named Dr. Cook and Dr. Vaughan, who realized that after exposure to a food that didn't cause an acute symptomatology, but was a trigger of these chronic type of symptoms, there would be a drop in the number of leukocytes, peripheral leukocytes. So to test for it, they would actually get a baseline count using the method extant at the time, which was to count using squares. Do an in vivo challenge, take blood again 3 hours later, and then repeat the count. And if there was a lower count, it was presumed that there was a sensitivity. Afterwards, an allergist in El Paso, Texas, named Black, looked at whether or not he could discern morphological changes in peripheral leukocytes fixed on microscope slides and exposed to test substances. And he found that there were changes in the cells that one can visually see. And this concept was picked up, that was in 1957. This concept was picked up a few years later by a pair of ENT allergists in St. Louis at Washington University. And they did a lot more work and published several papers, and they called this test using the microscope the cytotoxic test. And that was used enthusiastically by many people who followed Randolph and Cook and others who established this discipline of clinical ecology. The trouble was that the test was subjective, and you can imagine how tiring it would be to look at 100 or more slides and try and discern whether or not the cells have changed in some way, indicative of a response. So it was more or less discarded. And around that time, we started to investigate whether or not we can make measurements of the cells using objective electronic hematologic instrumentation and interface with computer and calculate the changes in the cells. And we started that work in the mid-'80s, and then in the couple of years we established a methodology and we started to market this test, and shortly afterwards we invited academicians and clinicians to independently evaluate it, and we went on the market, and here we are today. And every day, actually, we have samples coming into the United States lab from all over, with from physicians mostly, whose patients have not been helped by other issues, by other investigations. And this may, in some cases, be their last chance. And they present with all kinds of symptoms: rheumatology, chronic migraine, metabolic disorders, respiratory disorders. And now GI disorders have come into prominence through the work of many people, but particularly a friend and colleague named Alessio Fasano, who runs the Celiac Research Center at Harvard. And so I want to start sort of at the end with some of the most recent research that's been done in this area by Fasano, to put into perspective where we are right now, because really what we've seen is a great evolution, and I think it's going to continue, and we'll learn more and more about the operations of the innate immune system and how it mediates our relationship with the environment and can enhance health or could detract. About a year and a half ago, Fasano's group, working with people at the National Institute of Health Lymphocyte Biology Research Center and Leukocyte Biology Research Center, actually established that when gliadin, when an experimental animal is exposed to gliadin in vivo, the cells become activated, the neutrophils become activated. And it looks kind of like this. This is a video that Professor Fasano presented actually earlier than the publication at a conference that my company sponsored on gut, brain, and autoimmune diseases. And it's quite dramatic in showing quite graphically what happens or what can happen following a gluten challenge.
What you see here is the cross-section of this core of the villus. This black stuff here are the capillaries. And these green dots that you will see a little bit better are the soldiers, are neutrophils. They are particularly treated in these transgenic animals with a fruit fly gene that makes them to be fluorescent. Okay? So what you're going to see now is on the left, these are the villi of a mouse that has been exposed to another protein, BSA. This is the mouse that has been gavaged with gluten. And this is pretty much what you're going to see there. So see how many of these soldiers are stuck on the wall of the capillaries here, and then eventually start to move out and get into the external part, what we call the lamina propria. While this guy here, they keep traveling, and they stay in the lumen. So in the animals that was exposed to gluten, gluten is attracting the soldiers outside of the capillary, and they start to really get on the battlefield where gluten, and God knows again what else, is sitting in there because it's been, absorbed through this breach of the barrier. While these guys, they keep going. Again, I mean, who is close by may see that. But even if you're not that close, you see the mess that is happening here. And this is all due to the simple difference that here you have bovine serum albumin, and here you have gluten. That's all.
Just because this is perhaps just a better graphic, I'm going to play it for a few seconds. This is actually from the published paper. And you start to see the neutrophils. Again, this is a transgenic mouse with a gene transplanted in from a fruit fly, which will fluoresce when the cells become activated, like a luciferase reaction. So we know that the neutrophils, they'll distort their cytoskeletal and change, and they'll squeeze out through the capillaries into the tissue to attack pathogens in the tissue. So this is measurement of activation of neutrophil simply by exposure to gluten and looking at bovine serum albumin as a control. And same thing is seen there. And the same thing is actually seen in vitro. And the interesting thing here is in the top we have a control, PBS, it's a neutral solution, and it doesn't cause the cells to migrate. Here we have actually a substance which is synthesized, FMLP, and it's used in laboratory studies because it mimics the molecular configuration of the cell wall of gram-positive bacteria. So it activates neutrophils. So that activates the neutrophils in the same way that the gluten does. And what's kind of interesting about it is that these cells are doing this independent of another phenomenon, which we've all heard about, which is the breach of the intestinal barrier, or in common parlance called leaky gut. And there's a lot of controversy about whether leaky gut really exists, and so or whether it doesn't exist. And I'm told a documentary that aired a few days ago saying that it doesn't really exist, it's in people's imaginations, but it does exist. One of the other things about gluten, which I just alluded to, is that aside from being an activator of cells, and of course gliadin, which is part of gluten, is a very interesting molecule because we don't digest it. And it also contains a lot of proline and glutamine, which is just very resistant to pancreatic enzymes and brush border enzymes. So people who have the wrong genes can develop celiac disease because the gluten activates it. So in a sense, gluten has two roles here. One, and this is what I want to introduce, is this phenomenon that gluten induces. Gliadin fragments bind to a receptor on the surface of enterocytes. And remember, there's only a monolayer in our intestines, which serves as a barrier between us and the environment, a single layer of cells. And the system is very intelligent. It maintains tight junctures normally, so bacteria and undigested food can't come through and wreak havoc in the lamina propria, where 70 or 80% of the immune system is. But the gluten, aside from the fact that it's an allergen, also triggers the breakdown of these tight juncture proteins, and we get this phenomenon called leaky gut. But they continued the experiments, and they used a substance which blocks a certain receptor, FPR1 receptor, which recognizes bacteria, and it stopped the migration of the neutrophils. So it's interesting because it suggests that the body thinks gluten is a pathogen. And you can block that. Now, one of the interesting things about Alessio's earlier work was that he's credited with being the discoverer of zonulin. Now, can you guys tell me, have you heard about zonulin as the trigger of leaky gut? If you would nod or raise your hands. So it's not totally new? Is it totally new for anybody at all? Yeah. So anyway, one of the things that we found out is that zonulin was identified. And zonulin doesn't occur in animals. It's only occurring in humans. And humans are the only species that have autoimmune disease. And right now, the count is up to in the 80s in terms of the number of autoimmune diseases we have. So here's this connection between this ubiquitous environmental substance that we eat, that many people eat several times every single day, and the breakdown of the very delicate intestinal barrier and triggering and the rise that we see in autoimmune diseases. Drilling down a little bit further, the question was, is there, some people, for example, not a few people, about 70 million people in the United States avoid gluten, either restricted or just don't eat it altogether. That's a lot of people. What is that, like, 2.5 times the population of Australia. So do they all have celiac disease? Of course not. But people claim they feel better when they avoid gluten. So there's a distinction in the nomenclature where someone doesn't have the haplotypes to develop celiac, but they feel better when they go off gluten. We call that gluten sensitivity. And in trying to figure out what causes gluten sensitivity and what causes celiac, came up with some interesting findings. The findings were that the gluten sensitivity condition is mediated by the innate immune system. And I'm not going to get into this too much right now, because I'll get into a little bit. The way they figured that out is they looked at the transcripts for receptors on cells that only occur on innate immune cells, not specific immune cells. And they found that in the gluten sensitivity population, they're upregulated significantly as compared with the celiac disease population, and negative controls, who in this case are people who have dyspepsia. So the inference is clearly that gluten sensitivity, unlike celiac disease, is mediated by the innate immune system. Now, I want to speak in a minute. This is the last time we're going to hear Alessio tonight. This is also at the conference I sponsored. Talking about this somewhat controversial issue of leaky gut, and then kind of put that into perspective. So,
the second element that just came into the picture, that again I will briefly mention today, but we're going to go in more deep details tomorrow, is this leaky gut. Now, again, guys, I'm pretty sure that you're familiar with this leaky gut syndrome and so on and so forth. And again, unfortunately, there is a very few facts because this is a brand new field, a lot of fantasies, to the point in which, again, if you go Google leaky gut syndrome, it looks like that everything that goes wrong in the world is due to the leaky gut. So if we had, Hurricane Sandy was because of leaky gut. If the election next week would not go the way that you like, it's going to be leaky gut, and so on. Right.
So, yeah, that was actually just before the last election, the election before last. What can I say? There must be a leaky gut issue going on in the U.S. So, how do you test for intestinal permeability? You can look at zonulin levels in the serum or in the feces, right? And NutriPath does that. You can also do a functional test with lactulose mannitol. Are you guys familiar with that? You take a solution of 2 sugars, and lactulose is a big one, and it shouldn't transverse the cells, so if it's excreted in significant proportion to the control, which is the mannitol, which is a small sugar, which does enter the intestine paracellularly, going across the cell. Then the presumption is that there's a breakdown of the intestinal barrier, that there's leaky gut there going on. What's interesting that we found out, and this is again just speaking to the question as to whether or not leaky gut is real and who gets leaky gut, is that the controls, and again in this study they used people who had dyspepsia as controls, looked also at patients with celiac disease and gluten sensitivity, again defined as people who go off gluten and feel better but don't have celiac. So what's interesting is that the people who have gluten sensitivity actually don't have leaky gut. They have, certainly the people with celiac, they do, more of that lactulose, leaks in through the gut. So what's that all mean? In the same body of research, there was an investigation of what kind of different lab findings are associated with celiac disease versus gluten sensitivity, or negative controls or dyspeptic controls. What's interesting is looking at 42 patients with celiac, 26 people with gluten sensitivity, looking at the different symptoms. Gluten sensitivity and celiac kind of look a lot the same. There's a lot of other conditions that follow on from celiac disease, which you can imagine if your villi are decimated, you're not absorbing nutrients, and so there's going to be problems. So how are those problems going to manifest? Sensitivity to gluten also causes some weird problems. What do we see that's interesting about the differences? The first thing is that looking at a classical allergy test, and Peter and Mary asked me to kind of emphasize this point a little bit, so forgive me if I'm being repetitive, but we're not looking at allergy. And in this study, there was a RAS test performed on most of the patients. And out of the whole group of celiac patients, of which 18 were assayed, none of them had elevated allergen-specific IgE antibodies to wheat. That's not an allergy. None of the gluten sensitivity folks had it, and one of the controls actually had a positive. What's, I think, all interesting about the findings, which probably distinguishes gluten sensitivity from celiac disease, is the presence of tissue transglutaminase. Now, TTG, well, out of the 42 celiac patients, 37 had elevated TTG. None of the gluten sensitivity subjects did. None of the controls had it. The other strong distinguisher is the MHC profile, or the genetics. You're familiar with the genes that are tested for celiac, human lymphocyte antigens DQ2 and DQ8. Yeah, if you don't have those genes, you can't get celiac. And the patients with celiac mostly had expressed those genes. Some of the gluten sensitivity folks did, none of the controls did. The genes for celiac, like other genes that are associated with autoimmune disorders, are on the long arm, located on the long arm of chromosome number 6. And they encode for MHC molecules, MHC type 2 molecules. Now, why do those genes cause these problems? And again, celiac disease is a function of specific immunity. Now, specific means different from innate. Innate is the first line of defense. Specific is called, the specific immune system is called in to take over once the innate immune system defenses have failed. And they're very, very, can be very strong, obviously, as is in the case of celiac. So to trigger a specific immune response, unlike an innate immune response where we saw the neutrophils reacting to the gliadin presentation, both in the experimental animal in vivo and then also in vitro, that won't happen. You won't get the T cell or specific immune system response without there being an initial series of steps, mainly called antigen presentation, because the T cells can be so aggressive and set up so many different diabolical ways of causing damage that they just don't respond that easily. And only a few of them respond to a specific antigen. That's why it's called specific immunity. Now, they may signal other cells to produce antibodies, and they may become cytotoxic, and they may recognize markers on infected cells and start killing them, and so forth. But that all requires that first and trigger gets, in this case, in this graphic, which is an old graphic from Scientific American, if we could think of the antigen here as being gliadin peptides, they get taken up by the antigen presenting cell, which is found to be a dendritic cell. They get translocated to a lysosome, they're broken down, and then they bind with an MHC protein, a class 2. Now, what encodes for these proteins are those human lymphocyte antigens. So if this step doesn't happen, you don't get celiac disease. So if you don't have the gene, you don't manufacture this carrier molecule, which conforms with those peptides and then translocates the peptides to the surface of the antigen-presenting cell, where it gets noticed by a T cell that has that special kind of receptor. So all cells actually have MHC molecules so that this immune system knows not to go haywire on itself. Those are class 1, this is class 2. So the T cells get activated, and then they multiply themselves, and they may send out signals to B cells to multiply themselves or clone themselves, and express soluble receptors of the same molecular conformation, which then go into the circulation. And then we'll grab that antibody and hopefully prevent it from causing a problem. So that's specific immunity. Now, the ALCAT test doesn't test for that. And that, of course, would apply to allergy. Allergy doesn't happen through the innate immune system. Allergy happens through the specific immune system. And what determines whether we have an allergic reaction or another kind of immunological response, like in the response to gluten with active cytotoxic T lymphocytes, has to do, of course, with the messages that come across. So in allergy, the activated T cells might send a message, an interleukin-4, interleukin-13 message, and then the B cells are programmed to produce an isotype of IgE, a very rare antibody. And when that binds again to a mast cell or basophilin, is of course linked by the allergen another time, we get degranulation, histamine release, and all the sequelae that follow on. Now, if it's an interferon gamma signal, the isotype will be IgG. And then we get these antibodies that bind to, let's say, a food particle, and then kind of immobilize it, and then it can be eliminated by the monocyte macrophage system. And actually, that usually doesn't cause symptoms. IgG doesn't cause symptoms, IgE does. There's about 10,000 IgG molecules for every single IgE molecule. So the IgE molecule is the dangerous one. It's our defense against parasites. In fact, there have been attempts to try and increase IgG to prevent allergy. And when you increase IgG, you decrease IgE. When you increase IgG, you decrease IgE, and you decrease allergy. And so that's just the body's way to get rid of excess antigen from the circulation. So I was asked to speak about that as well. So any further questions, we can kind of go through that. Now, coming back to celiac, we have here in our graphic, again, the monolayer of enterocytes. And we have here a gluten peptide triggering from the enterocyte the release of zonulin, which again binds to that CCXR receptor and opens up the tight junctures. And we get gluten fragments coming in. Now, the gluten fragments, the enterocytes also send chemical messages to T cells, which causes them to migrate up into the lining, and that's a dangerous process. And it damages the enterocytes, and the enterocytes respond by producing an enzyme called tissue transglutaminase, which we looked at a few minutes ago. That's a product of damaged enterocytes. But what happens then is kind of interesting. The TTG binds to the gluten fragments and then gets picked up by our old friend, the dendritic cell. Now, if this dendritic cell belongs to a person who has those MHC molecules based on their genetic makeup, or in their chromosome 6, that recognize this and present it to a T cell, then the T cell gets activated and sends out chemicals that are toxic to enterocytes, turn on killer T cells, and might switch on B cells to produce antibodies. But it says here that B cells release antibody molecules targeted to gluten and TTG. They might cause further damage when they hit their targets on or near enterocytes, but the role of antibodies in the disease is unclear. And that's another point. It's the cells, in this case the T cells, that are actually causing the damage, not the antibodies. The antibodies can't do that. Antibodies can only cause damage if they activate a cell. And the cell causes damage by releasing free radicals and toxic mediators, prostaglandins, leukotrienes, and all kinds of nasties. And of course, before all this happens, the person may not have this activation of the T cells because they don't have the genes to produce the molecule that presents the antigen to the T cell. And so this whole side of the equation just doesn't happen if you lack the genes, but you could still get irritation or gluten sensitivity or other gastroenteropathies, IBS, which are mediated, as we saw before, by the innate immune system. So it's good to, if you want to really drill down and find out if a patient has risk for celiac, it's good to look and see if there's a disease process going on by looking at TTG. If you want to know if they're at risk for celiac, even if they don't have elevated TTG, look at their genes. If you want to look at whether or not it activates their innate immune system and causes some... Now, it's interesting that over time we see an increase in autoimmune diseases. This paper from New England Journal of Medicine talks about how risk for autoimmunity goes up following infections. And it's kind of interesting that the molecule on the neutrophils that got activated by the gliadin peptides was the same molecule that recognizes pathogens. So there might be a link there. Again, there are many disorders, and there's been autoimmune disorders. There's been genes associated with those, including type 1 diabetes. This was a paper that came from Cold Spring Harbor, which is actually a beautiful place where James Watson was the director until recently, and it's right near where I grew up. It's a beautiful bucolic area. And they looked at the increase, and again, we see that there is an increase in type 1 diabetes. They looked at the increase and saw who's getting more type 1 diabetes. Is it people with the medium genotype risk? No, the low-risk people had a greater increase in type 1 diabetes, and the people with the very low risk had the greatest increase. It makes you wonder. So there's the interaction between the genes, certainly, that can, through certain pathways, like the one we reviewed talking about celiac, and others that are probably not specific immune, but innate immune, that are causing an increase in autoimmune diseases. And that's where another area where ALCAT is very useful, because people come in, some of them have MS, some of them have less pernicious conditions, migraines, skin rashes, respiratory issues, irritable bowel, and so forth. But some come in with these really difficult, debilitating diseases, and there's no relief. And then they change their diet, and then those symptoms go into remission. So it's very interesting and subtle what this innate immune response can do. When you look at IgG, and you really, if you can get people, they feel better by following the results of an IgG diet. But they feel better if you give them a placebo diet. And I know that. I mean, it's known. And we did that research recently with folks at Yale, where we looked at patients with IBS and we gave them an ALCAT diet or we gave them a placebo diet. And both diets were equal in terms of rigor and complexity. So if a person had an ALCAT test and it showed that they were reactive to 3 meats, 4 vegetables, 2 legumes, a fruit, a nut, 1 dairy product, then for the control group, and everybody was blinded there, we gave them a diet which eliminated 3 meats, 2 vegetables, 2 legumes, et cetera, the same thing, but they were the wrong ones. Now, they got better, but not as much better as the control group. So typically, if people want to assess the sensitivity and specificity or the efficacy of an in vitro test, you have to compare it to a known standard. And the only known standard for a food intolerance is a feeding challenge carried out double-blind and hopefully placebo-controlled. And when you do that with an ALCAT test, you get correlations very high, 80s and 90%. Now with an IgG test, you don't. You get some response to treatment, but probably 2 reasons for that. If a person goes off the foods that they're eating the most of, well, you might hit it for the right food for the wrong reason, or you get a placebo effect, so forth. So I hope that covers that. Other researchers have found that you can have inducement of neutrophil activation with other foods. So in 18 out of 20 patients who responded to a gluten challenge, 10 of them also responded to a casein challenge or a milk challenge. And the same group published that a number of them responded also to a corn challenge. So there's clearly other pathways besides just this one where you have the celiac disease. So I thought I'd include something which comes from a clinic in Germany where they also do an in vivo study with humans to look at intestinal permeability induced by foods by gavaging the person with the different allergens. Now this poor chap had all kinds of issues going on, and that's what he looked like, kind of puffy, eczema around the eyes, terribly fatigued and all that. It's in German, so I'm going to translate a little bit. They took blood and they looked for allergen-specific IgE antibodies. They didn't find it. They scratch tested him. They didn't find any answers. So they brought him in and they did kind of an invasive thing. I wouldn't want to have to do this, but it worked. They tested a number of different allergens and looked on a screen. There's the guy with the magic wand. Looked at the screen to see if gavaging with individual foods actually would open up the tight junctures as a marker for, intolerance. And in this case, they looked at soy, milk, wheat, and what they found at the end of the day was that, in his case, yeast was a problem. Now, he's German. I have a lot of German bread, drink a lot of German beer, so it's not terribly surprising. So you can see when they carry out the study, no reaction to the milk. The light's not shining through the intestines, as we'll see in a moment. And when they go with the yeast, there's the milk, it's negative. And then with the yeast, it's positive. Half is German for yeast. There you go. So, I'm going to skip over a couple of different things. It's interesting, we talk about milk a lot as being reactive. And, the latest, I think, cutting edge right now is really looking at how our genes respond. And, we have a genetic condition. We all know that you might be prone towards something, but you don't express it. You don't fall to that. And it depends on how we express those genes. In fact, in the U.S., the government has stated that 70% of chronic disease is preventable through lifestyle. And I came across this study recently, which I thought was kind of interesting, where it shows that unboiled milk or human breast milk actually unmethylates the promoter region of genes which are associated with the factors that upregulate Treg cells, which are anti-inflammatory. So milk kind of gets a bad rap, but I don't think it's such a bad food. I think when you pasteurize it, you denature the proteins. If you sort of feed the cow corn and soy, which contain a lot of omega-6, shoot up the cow with antibiotics, growth factors, the product is not really milk anymore. But human milk is really healthy, and they identified 6 ways in which it helped to prevent allergy. And of course, introducing inoculation of beneficial bacteria and providing food for the beneficial bacteria, but also, upregulation of this FOXP gene, which differentiates cells, naive T cells, into regulatory cells that are anti-inflammatory. So there's a lot to this. This is one of the studies from London showing correlation of alcohol with double-blind challenge. As Peter started to mention, in our company, we're an FDA-registered device manufacturer as well as a laboratory. So we build the instrument according to what our needs are. And this is a graphic of our next generation instrument. It's our 5th generation of instrument. It's a cell counter. And how it works, so this is the method how we count cells. So we want to, again, look at how a cell responds to a challenge, ex vivo, to foods or whatnot. So it's a very simple principle that was actually invented by a man named Wallace Coulter. And the Coulter counter was named after Wallace because he built the Coulter counter. And essentially, it's very simple in concept. You take these, let me put it this way. There's a story that Wallace had a job when he was in his 20s counting fish. And one day he had the bright idea that if he narrowed the channel where the fish would swim through such that only one fish could go through at a time, and then rig up an electrical current so when the fish passed through it would impede the electrical current and trigger a bell, he could just kick back and just listen for the bell. You didn't have to look very carefully. And so he got the idea to apply that principle on a microscopic level to count particles. So the idea is to suspend particles in an electrolytic solution with defined electrical conductive qualities and put electrodes on either side of a channel and let the particles run through there. And of course, you have to control the flow rates very carefully and control the electrical voltage very carefully and all that. But if you do that, when the cell goes through, it impedes the current in direct proportion to its mass. And, whoops, sorry about that. And then you're able to do something like generate the counting of cells in a number of different size channels and create a size distribution curve or histogram. And that's what we do. So we run a number of samples that don't have any test reagent, and we average those, and that's our baseline. And then we take the same blood, treated the same way, except exposed to a substance, and run it through and get a size distribution curve for that. And then plot one against the other. And if there's been a change such that the cells have decreased in numbers because they've actually degranulated, they've released granules that contain toxic stuff and go out and cause problems. They decrease in numbers, but before that actually happens, they increase in size. Before that happens, they decrease in size. So we've kind of mapped out those steps and run our assay accordingly. And then if we get a reaction to a food, it would look something like this, where there's actually a decrease in the number of innate immune cells. These are the larger cells, these are the lymphocytes. And some of them are still not dead, but they've swollen up. And then you just do some calculus and figure out how strong the reaction is and report it out. So if you look at that kind of phenomenon under a microscope, if you have a cell that has no reaction, it stays looking nice, and the graphs line up and you don't see a dislocation, and it's verified on flow cytometry. If you get this kind of swelling, there's all kinds of things happening internally that the people with the microscopes talked about, and now we understand much, much more. You get this kind of dislocation in the curve. And then if it really goes to an extreme where there's total degranulation, you'll have, cells are missing, they're gone. So, again, Peter and Mary asked me to kind of, again, talk about when should you order this test. And some of the results from a study done at Baylor looked at a number of different conditions and saw that there was improvement reported by the patient in things like, psychological, tiredness, cravings, tension, fatigue, stress, cravings for sweets, so on and so forth, arthritis, eczema, respiratory conditions, and so forth. In a study in Pavia, this was the outcome looking at an elimination diet based on ALCAT. 71% had very important improvement, and the majority, only a couple of percent didn't respond. This is a report from the recent study at Yale, looking at IBS and comparing it to a control group that I described, and the changes in symptoms. Accepted scales, these GIS and SSS, are used to assess the severity of symptoms associated with IBS. And the placebo group improves and the control group improves much more. I put together, and this could be hundreds of slides, conditions where circulating neutrophils or activating neutrophils are implicated in neurodegenerative diseases. We've talked about that. Some of the other work at Yale, and I apologize, I last gave this presentation in Germany and I forgot to change the title, but that's the title. ALCAT test predicts the release of DNA by innate immune peripheral blood leukocytes via protein kinase C dependent pathway. So when the cells degranulate, they release DNA, and that's not such a good thing, and much more so in a positive food than in a negative food. And we found out that it's dependent on protein kinase C and NF-kappa B by blocking those pathways and seeing the inhibition of those responses. And it's understood now that the innate immune cells not only will release free radicals and toxic mediators, even after they're dead, they release these things called NETs, neutrophil extension traps. And that's a dead neutrophil with neutrophil extension traps trapping bacteria. Now, the interesting thing is that even though this is dead, all the stuff inside didn't really disappear. So those nets are comprised of cytoplasmic content and nuclear content. And the DNA is very toxic to the bacteria, and that's why the immune system implements this as a strategy against pathogens. There's a net trapping Klebsiella. That's a good strategy to kill these things. But if you're doing it all the time with food that you're eating every day, you're overdoing it. And the problem is this DNA is toxic. It should be inside the cells, and it does cause problems. It kills these pathogens. It's also damaging to us, and it is associated with autoimmune disease. At least it's been associated with lupus and arthritis. So that becomes more prominent depending on a person's genes and their ability to break down these products, DNAs, and so forth. And if it's dysregulated, then the other macrophages that would take it up are overwhelmed, and we get autoimmune disorders coming. It's proposed by the guys at Yale that the DNA released by peripheral blood leukocytes triggered by foods may be a mechanism that supports the rationale for why we see such a broad array of clinical improvement, any part of the body. The eosinophiles are a class of granulocytes that are involved a lot in the ALCAT reactions. Basophiles are not involved so much, and by the way, that's probably a reason why you shouldn't order an ALCAT for someone with a known allergy, because it won't work, because the basophiles are the cells that are involved in type 1 allergy, and they're only about 1 or 2% of the peripheral blood. So we don't pick those up because the signal to noise is too small. Eosinophils contain a lot of toxic granules. There's a list of the cytokines and chemokines and cationic proteins that are there. And if anyone doubts how serious it can be when you kind of irritate these cells and get them going, look at what happens to this poor worm, okay? I mean, they just demolish this. These are just cells just taking hold of this big creature and reducing it down to nothing. So that same kind of activation is happening sometimes when people eat a kiwi. In my case, this kind of thing happens if I have cucumbers. So it's really weird, but all foods contain chemicals that are toxins. I mean, that's how they protect themselves. And they also produce a lot of beneficial chemicals, but they have these toxins that can cause problems if we don't have the genes to detoxify them adequately. So again, lots of different things, even infertility in men, activated immune cells, damage to telomeres, which causes premature aging. Inflammation can cause oxidation of LDL, and it's a better marker for cardiovascular disease. When you look at CRP in addition to LDL, you get a linear increase in risk when you add the inflammatory component. After a heart attack, the damage to the heart muscle is caused by the neutrophils during the reperfusion phase. There's more zonulin present in people who have active MS than MS in remission. There's 2 to 8 times an increase in neutrophil superoxide production in patients with arthritis. This research is done from the Public School of Health at Harvard, and they found, at least in mice, that it's normal to have some kind of inflammation when there's over eating, particularly the carbs and fats. And there's a mechanism that counterregulates that. But if the genes for that mechanism are deficient, then you get metabolic syndrome. And one of the sequelae of that is the increase of various cytokines that are inflammatory, like IL-6. But what also is very interesting is you get an increase in this molecule in the visceral adipose tissue, white adipose tissue, called haptoglobulin. Anybody here familiar with haptoglobulin? Haptoglobulin's only known effective value is to bind and help rid the body of hemoglobin from dead red blood cells. But remember I said earlier that Alessio Fasano identified what zonulin is? And do you know what it is? It's prehaptoglobulin. Prehaptoglobulin 2. It kind of comes in two flavors, number 1 and number 2. And if you have the genes to make number 2 and you have inflammation, you have more of this haptoglobulin, so you have to have more prehaptoglobulin. So that means you'd also have more zonulin. So vicious cycle there. Very interesting that these things kind of come together. So this was a conclusion stated as the last sentence in the paper that I presented from Fasano about the neutrophil activation. And he's saying, what does it mean? Let's try and figure that out. And of course, the immune system is very subtle, it's very nuanced. It should be in balance. This is the cover of Science magazine from January 13. And this is a series of statements about how the immune system is both yin and yang. It gets activated and causes damage and protects us, but also reverts us back to homeostasis. So that's what we want, and that's what LCAT is used for, to figure out what our immune system likes and doesn't like. So that's my conclusions, and thank you very much for your kind attention.